So guys, now we are going to move onwards and see other metalloproteins which are responsible for oxygen carrier transport in the biological system. So hemocyanin is one of them. From the name, it feels like there is a heme group and there is a cyanide group, but no, there is not. There is neither heme nor cyanide over here. The hemocyanin molecule actually looks like this. This is the oxyhemocyanin. It actually has copper. It has a very simple structure as compared to heme. It does not have, contains no heme unit. Okay, so hemocyanin is a respiratory protein containing two copper centers at the active site. Very simple. It is a dioxygen carrier suspended in the hemolymph, that is the blood of most mollusks and arthropods. These are actually some classes of organisms, mollusks and arthropods. Also, in the deoxy form, if you want to see the deoxy form, it is actually looking like this, that the two copper centers are a little bit far away from each other. They're in the +1 oxidation state. They are held by nitrogen of the histidine groups, like this histidine side chain of the amino acid. All right, when it catches the oxygen, it is converting the oxygen into a peroxide, and then there is going to be a bridge like this. But how is this oxygen bound? Now copper +1 goes to copper +2. This was actually colorless. The deoxy form was colorless and the oxy form is going to be blue in color. That is because the copper 1+ is colorless and the copper 2+ is blue in color. All right. Now, yes, it is actually going to bind in this way. इसको हम क्या बोलते हैं? Mu peroxo bridge. That is bridging, but in a peroxo form. Mu peroxo bridge. Okay, this is what it is. The deoxy form contains copper 1 ion and is colorless, whereas the oxy form contains copper 2+ and is blue in color. Okay, very simple. In deoxy form, each copper 1+ is coordinated to 3 histidine residues of the amino acid. Side-on bridging coordination occurs with the dioxygen in the oxy form. तो ये जो यहाँ पे oxygen है, this is a side-on overlap. And many a times it is mentioned as eta2 or mu eta2 O2 2-, इस तरीके से लिखा जाता है. Why is it written like this? Mu is for bridging, eta2 is for hapticity, that on each side this dioxygen is showing a eta2 hapticity on this side also and on this side also. So it is a peroxide, O2 2-. Why is that so? Because when you are having copper 1+, you are having the dioxygen trying to connect to it. So each copper is going to give it 1 electron. तो एक इलेक्ट्रॉन दोनों ऑक्सीजन पे आ गए, then it becomes O2 2- and the copper becomes Cu2+ like this. That's why it is there in the peroxide form. Now here I am just giving you an example of octopus. Octopus, you might have seen, has copper based blue blood. Absolutely, the blood is going to be blue in color because it is having a hemocyanin. Other examples that have blue blood again are lobsters, crabs, pillbugs, shrimp, crayfish, scallops, barnacles, snails, small worms, except the earthworm, it has red. I guess it has hemerythrin. Clams, squid, slugs, mussels, horseshoe crabs, most of the spiders. All of these species, they fall under the category of arthropod and mollusk, and they have blue blood. Okay. Now we are going to move onwards and we talk about hemerythrin as well. So again, this is a metalloprotein, but again, hemerythrin ऐसा लगता है कि heme है, but there is no heme. This is a non-heme oligomeric respiratory protein of marine invertebrates, specifically marine invertebrates. All right, it transports oxygen just like hemoglobin does. All right, the two ions are coordinated to the protein through carboxylate side chain of 1 glutamate, 1 aspartate, and 5 histidine residues. This is telling you about the structure. So there are 2 iron ions, Fe2+, which are connected to each other via the acidic groups, the carboxylate groups of aspartic acid and the glutamic acid. And there's a slight difference between the structure of aspartic and glutamic acid. For example, This is your simple amino acid. All right, alpha amino acid ऐसा ही होता है, same carbon, 2 groups. Now on the side chain you need to have another acid. When it is CH2COH, that is going to be aspartic acid, and when it is CH2CH2COH, just one carbon extra, that is going to be the glutamic acid. So these two iron ions are actually connected by the glutamic acid, the glutamate of the side chain, not the main chain. Okay, because this is used up in bonding the whole peptide, forming the whole peptide. So it is the side chain which is actually holding these two iron ions together. Okay, and there is one more bridging hydroxyl group, as you can see, and there is a difference between the coordination state of iron. Here it is going to be hexa coordinated, all right, and the lower one is going to be only penta coordinated. And this is what you see in the deoxyhemerythrin. Deoxyhemerythrin. All right, this is it. Now let's see more important points. In the deoxy form, one iron is hexa coordinated and the other is penta coordinated. Both of these are still iron 2 plus only, and it is still a colorless molecule. The iron 2 plus ion transfers one electron each to make Fe3 plus bind with oxygen. So again here basically dioxygen is carried in the form of a peroxide, just like hemocyanin. Peroxo linkage banta hai, that forms a bright reddish violet color in the oxy form. So that is the color of blood where hemerythrin is present, red violet color. Okay. In oxy form, both the iron 3 plus are hexa coordinated. So let's see. So what happens is both of these iron ions give one electron each to the oxygen. Then it becomes O2 2 minus. That is going to be your peroxo. All right. And this is how it will connect. One to the iron, one oxygen to the iron. The other oxygen does not connect to that iron. It's very far away. So it is held on by the hydrogen bridge. It is held on by the hydrogen bridge. All right, very well. Also, what is the magnetic property if anybody asks? Then in the oxy form, you will see both of the iron are 3 plus, D5 system. All the 5 electrons are unpaired, but they will antiferromagnetically couple. Antiferromagnetic क्या होता है? The spins are opposite, just that. So they are diamagnetic. They are diamagnetic to each other. All right, what about this? Fe2 plus 4 unpaired electrons, same over here. Again, antiferromagnetic coupling can give you diamagnetic. So, no cooperativity effect shown here, neither in hemocyanin, because it's a very simple oligomeric protein. It's not like a big protein just like hemoglobin, which is a tetramer. Okay, this is only a monomer, and that's why no cooperativity effect shown. Unlike hemoglobin, less affinity towards carbon dioxide or carbon monoxide due to the hydrogen bonding. It's very simple, and there is no affinity for both of these poisonous gases also. Hemerythrin typically exists in the form of an octamer with a molecular weight this much. Exists in the form of an octamer, matlab ki 8 units are going to stay combined, okay, of hemerythrin, like that, right? So this is it about the hemerythrin, and now we will do some comparison. Okay, वो last में सब हम compare करते हैं so that we remember what we have studied. Now some properties of hemoglobin, myoglobin, hemerythrin, hemocyanin. This is the most asked topic. All right, the maximum PYQ of bio-inorganic you will see from these topics only. So first one, oxidation state in the deoxy form. Hemoglobin also +2, myoglobin +2, hemerythrin +2, hemocyanin +1 क्योंकि कॉपर है इसीलिए. Metal oxygen कैसे bind करता है? One iron atom takes up one oxygen, one dioxygen, that is. In myoglobin same, but hemerythrin में 2 iron atom will take one oxygen molecule. Got it? That is, in the hemoglobin there's only one porphyrin ligand that is going to take up this dioxygen, but hemerythrin में, you know, there are 2 iron ions which is going to both collectively form a molecule to take up one oxygen. जबकि इसको बोला ऐसे ही जाएगा कि 1 unit of heme can take one oxygen molecule and 1 unit of hemerythrin, and hemocyanin can also take one oxygen molecule. Okay, it can carry like that. Now we move onwards. Color of the deoxyform. हेमोग्लोबिन का कलर होता है रेड पर्पल, इट्स रेड पर्पल कलर. मायोग्लोबिन कलर होता है डल पर्पल कलर. हेमेरिथ्रिन is colorless, hemocyanin is also colorless, लेकिन in the oxy form it is going to be red, myoglobin again bright red, hemerythrin में violet pink, alright, it's like reddish violet color, and hemocyanin is blue. Okay, just remember this. Metal coordination, absolutely. Macrocyclic ligand, porphyrin ring, the same in the myoglobin. लेकिन हेमेरिथ्रिन में ऐसा नहीं होता है. Side chains, whose side chains? Glutamic acid and aspartic acids. So protein side chain, glutamic acid and aspartic acid both, as well as histidine nitrogens. Okay. Again, hemocyanin protein side chain, but here only histidine are bound. ठीक है. Molecular weight कितना होता है? 65,000 units. 16.7. This is 108 because this exists as an octamer. That's why so much. And hemocyanin शायद और भी ज़्यादा को मतलब collect होके रहता है. I don't know how many, but yeah, from here, looking from the molecular weight, it looks like that there a lot of small units are setting themselves together to make a bigger unit. Okay. Number of subunits 418 octamer, as I said, and this is many. That's why so much high molecular weight. Okay. So even though the structure of hemocyanin is the easiest, but there are many subunits which are like sticking together and making a bigger molecule, bigger system. Alright, I hope that this makes a lot of things clear about these 4 oxygen transport systems. Now we are going to do some really important question which will make the topic even more clear. So first thing is iron 2 plus porphyrins, they fail to exhibit reversible oxygen transport and cannot differentiate carbon monoxide from oxygen. However, hemoglobin is free from both of these pitfalls. Among the following, the correct set of statement is. So understand this question, what the questioner is asking you. Once you have understood, it's very easy to answer. Try to stop the video and then do it by yourself. Alright guys, so you must have figured out this question. Now, what is the correct sentence for the deoxyhemerythrin and deoxyhemocyanin? Both of these are asked in the deoxy form. They are involved in the oxygen transport in biological system. That is correct. They contain 2 metal ions in their active site. That is also correct. Active site metal centers are bridged by amino acid residues. This is only correct for hemerythrin. This is not correct for hemocyanin, hemerythrin only. So I would say this is not correct. They prefer to bind only one oxygen per active site. This is also correct. That is, at one active site only one oxygen can be transported in the form of the peroxide. So A, B and D are the correct answer. This one, first one. Okay. Next question. Match the items given below in these 3 columns. So first you have to see metalloprotein. We have oxymyoglobin, oxyhemocyanin, oxyhemerythrin. Then species coordinated to metal centers. The way with which they are connected, coordinated like this. Resonance Raman oxygen OO stretching frequency, centimeter inverse. This is the one thing that I've told you for hemoglobin. So you kind of have an idea about this, right? So what are the correct matches? This question is going to take time if you are doing this for the first time, but it is an awesome question. Okay. So I will start answering. I hope that you just stop the video and do it by yourself. First thing, whenever oxymyoglobin is given, you are just going to recall whatever you know about oxyhemoglobin because the binding is almost the same. Okay, so we know about oxyhemoglobin that it has iron connecting to oxygen in the form of peroxide. So this peroxide, superoxide over here, this is going to be connected to iron in hemoglobin. So A matches with 3, which is going to match with 1100 centimeter inverse in the resonance Raman. All right, so this was the frequency that I told you for a superoxide, right? Now imagine if you have a superoxide that has a bond order which is less than 2 but greater than 1. So it is like a 1.5 something. Okay, so if the bond order is somewhere around 1.5. Now when you are increasing the electron density, let's say I'm writing peroxide, that is going to have a bond order of 1. Now both hemocyanin and hemerythrin are going to have a bond order of 1 only. Both of these are carrying peroxides only. But hemerythrin still has the um, oxygen, dioxygen bound with a hydrogen bond. So it actually looks like this. This is going to have a higher stretching frequency as compared to oxyhemocyanin, where it's not only a peroxide, it is a bridging peroxide and it has bound to both the metals. So because of a lot of back bonding, the bond order decreases further. So this is going to have the lowest bond order and the value of 803. Okay, this is how you have to match. Oxyhemoglobin maximum frequency A matches with 3 and Z. So only one option A, 3 and Z. Then B, 1 and Y. B, 1 and Y. Then lastly C3, C2, X4. Yeah, so A is your correct answer. Okay, just go to the structures, you will know. Active site structure for oxyhemerythrin is, yeah, so oxyhemerythrin ka direct structure, right? Just stop the video and figure out which is the correct structure. First thing, both of the iron cannot be pentacoordinated, so this is wrong. One of the iron has to have 3 histidines. Again wrong. Here 3 histidine and this one 2 histidine. So these two are looking correct. Now which one of these is correct? Now in the oxyhemerythrin, both the iron will be in the +3 oxidation state and not +2. This is +3 but this is +2, so this is wrong. Both of them are actually giving 1 electron each to the dioxygen molecule to kind of convert this into peroxide. Okay, so C is the correct answer. The other things that you have to check, that to the coordinated iron, the dioxygen will be connected and one hydrogen bond is there. Actually the hydrogen bond is supposed to be like this. Okay, anyways, that's a minor thing. And both of these are connected through the carboxylate residue of glutamate and aspartate, right? So C is the correct answer. Next, bleeding stops by the application of ferric chloride. The reason being, okay, so whenever we have a cut, we apply ferric chloride or some other salt. Why does the bleeding stop? Very common sense question. Okay, so let's answer. The blood reacts and forms a solid that seals the blood vessel. Okay. Blood is coagulated and thus the blood vessel is sealed. Ferric chloride seals the blood vessel. It cannot directly seal the blood vessel. Interaction between ferric chloride and blood results in lowering of the local pressure and hence the bleeding stops. This is wrong. Actually, blood is coagulated. Blood is a colloid. Okay. And whenever you put or whenever you introduce an electrolyte to a colloid, it is going to coagulate. So ferric chloride is an electrolyte. It's an ionic compound. As soon as you bring any ionic compound, you can also take alum or any other electrolyte, right, which does not burn. So whenever you are having a cut, even if you put alum over it, the blood is going to stop because of coagulation. The coagulation takes place. Okay. Now, next PYQ. Complexes which has or have unpaired electrons that is equal to that of iron center in oxymyoglobin is R. That is, which of these complexes will have the same number of unpaired electrons with respect to the iron center in oxymyoglobin. See, the questioner is not directly asking you oxyhemoglobin, but the the type of bonding in oxymyoglobin and hemoglobin we consider the same. So it's the same thing if they ask you oxymyoglobin or oxyhemoglobin. It's the same thing. Okay, so yeah, try to answer this question. We consider that oxyhemoglobin has iron 3 plus in the low spin. That means it is going to have 5 electrons like this. Okay, one unpaired electron. So considering all of these complexes, oxalato is a weak field ligand. It is not going to make this a low spin complex. So this is definitely iron 3 plus, but it is going to be high spin, all 5 unpaired electrons. Next, this is iron 3 plus. This is going to be low spin because of the cyanide. And here it is going to show you one electron just like this one. So B is correct. Nickel tetrachloride. So nickel is present in +2 oxidation state. That means it's a D8 system. Nickel 2 plus D8 system. It's going to be high spin, but वैसे D8 is not going to show that kind of a high spin, low spin possibility. It is always going to be like this. So there are going to be 3 unpaired electrons. No. D option, copper 2 plus is a D9 system, tetrahedral or square planar. This is actually square planar complex, and it is going to have one unpaired electron, because in a square planar complex, this is how you're going to split 1, 2, 3, 4, 5, 6, 7, 8 and 9, one unpaired electron. So D, B and D are correct. This is the correct answer. Only one unpaired electron we want. Okay. Next. Under physiological condition, oxygen is binding to deoxyhemoglobin and deoxymyoglobin. The binding curve and the pH dependence respectively are. This was asked in December 2014 for CSIRNET. Very interesting question, guys. Stop the video, do it by yourself. So you have already seen this. The hemoglobin has a sigmoidal curve. Hemoglobin has a sigmoidal curve because at the lower partial pressures of oxygen, it is not so keen to bind oxygen. However, myoglobin at lower partial pressures of oxygen is quickly going to take up oxygen from hemoglobin, right? So, this is hemoglobin is going to be sigmoidal and it is pH dependent. That is what is known as the Bohr effect, isn't it? pH dependence को ही तो Bohr effect बोलते हैं. And deoxymyoglobin is hyperbolic and pH independent. That's correct. Yes, sigmoidal and pH dependent on this. Alright, very simple. Chalte hain. Yeah, this is how it is. I've already shown this to you in the beginning of the class. The number of histidine amino acid nitrogen atom coordinate to bimetallic active site of oxyhemocyanin and oxyhemerythrin respectively are. That is, just see the options, how they are being asked. It's very simple. Okay, very simple. Try it out. All right. Basically, it's like this: aspartate, glutamate. So basically, the question is asking you how many histidine amino acid nitrogen atoms are present. So for hemocyanin, since it is asked first, it is going to be 3 and 3, 3 and 3, and for iron, it is going to be 2 and 3, or 3 and 2, whichever you want to say, but the answer is going to be option number B. It's very simple. In vitro reaction of an excess of oxygen with free Hem B in aqueous medium, and the end product is going to be. In vitro क्या होता है? In vitro means in the lab, and in vivo or vivo, that is basically in the body. Okay, so in vitro means in the lab. So when you are doing a reaction of excess of oxygen with a free Hem B group, Hem B means the hemoglobin heme, where you are having the iron and the porphyrin, right? That, only that, not the globin. So in the aqueous medium, the end product is going to be what? I think you can answer this really well. It is absolutely going to be hematine, because that's the irreversible mu oxodimer which is formed, which we have just studied in this session. Okay. Which one of the following statement for hemoglobin is not correct? Okay, yes, guys, there are 4 options. Not correct is asked. Read it out. Alright, so the binding with oxygen is weaker in comparison with myoglobin. Alright, this is correct. Iron is penta-coordinated, 5-coordinated. That is also correct, because one space is like for vacant for the oxygen, which is actually having a loosely bound water molecule that can be quickly replaced by oxygen. Okay, so we consider it penta-coordinated only. Iron is coplanar with the porphyrin ring in the absence of oxygen. No, in the deoxy form, it is not coplanar. So C is the not correct one. Oxidation state of iron is +2. Yes, in the deoxy form. Alright, so C is the correct answer. Hemocyanin contains, alright, hemocyanin contains what? Now here it's not mentioned whether it is deoxyhemocyanin or oxyhemocyanin. You have to figure it out based on the options. So yeah, read them out. Based on the language of option, let's see. So a dinuclear copper core binds dioxygen in the cuprous state. Cuprous is the +1. A dinuclear copper core binds dioxygen in the cupric state, that is cupric +2. A mononuclear तो है ही नहीं. It's not mononuclear because it is dinuclear, right? So either A or B. Which state it is actually binding the dioxygen? In the +2 form, isn't it? So B is the correct answer. In the cupric state, you are going to have the oxy form of hemocyanin. Okay. Now, very interesting question over here. Match the following. Now, definitely, guys, we have not yet studied about the other things, but we will do very soon after this. So, part A and part B, you have to match and then select the correct codes. Okay. Yeah, just stop the video, do it by yourself. Both the myoglobin and hemoglobin are given. Okay. So vitamin B12, carboxypeptidase, this is S, ferredoxin. So myoglobin is actually oxygen storage metalloprotein. It matches with C. One matches with C. There's C over here too. Hemoglobin is the oxygen transport protein. Two matches with D. So this is wrong. Vitamin B12 does the group transfer reactions, and it has cobalt definitely. So 3 matches with A. Carboxypeptidase is a peptide hydrolyzer, absolutely. So 4 matches with E. Ferredoxin is going to be an electron transferring agent. So this is the correct answer, option number A. Alright, next question. Hemoglobin is an oxygen carrying protein. The correct statement about oxyhemoglobin is that. So you see there are so many questions which are asked on the oxygen transport. Yes. Read out the options. Do it by yourself. Correct statement about oxyhemoglobin. Chalo. Metal is low spin in +3 oxidation state, while dioxygen is the O2 minus form, that is the superoxide form. Alright, this seems to be correct. Metal is high spin. No, in the oxy form it's not high spin. Metal is low spin in the +3. Yes. Dioxygen is the neutral form. No. Metal is high spin. Bilkul galat. Only A is the correct answer. That's it. Now, this question, very important one. The red color of oxyhemoglobin is mainly due to. I told you this before also. Just try it out. It's a very interesting question. Is it because of the DD transition? Metal to ligand charge transfer, ligand to metal charge transfer, intraligand pi to pi star transition. Why do we have the red color? It is because of the intraligand pi to pi star transition, red color, which falls into the same 400-500 region. That is because of the solid band, and that's a property of the metalloporphyrin. Why do we have the color, red color on oxyhemoglobin? That is because oxyhemoglobin has the iron into the plane. So iron becomes coplanar with the ring, and the ring is also not tensed anymore. It is like relaxed, and in the relaxed state, there is a better pi to pi star transition. That's why the intense color. So D is the correct answer. Last question, oxymyoglobin, MbO2, oxyhemoglobin, respectively are. What is the magnetic property? Yes, very interesting. In the oxy form, see, it's the same thing. Iron in the +3 oxidation state having O2 minus superoxide. So there is one unpaired electron on the iron and one unpaired electron on the oxygen. They are going to be antiferromagnetically coupled. So diamagnetic and diamagnetic. Sorry. Diamagnetic and diamagnetic, both of them in the oxy form. Okay. So yes guys, I'm going to see you all soon in another video. Just go through all of the things that we have studied and please make notes. Alright, I'll see you all soon in another session.